curriculum. Also we continue with this, with the second part. We're going to look for the mitochondria and Golgi apparatus, guys. Now, starting with the cell membrane, what do we need to know about the membrane? This is the outermost layer of the cells, which actually define the shape of and also the border cells. The cell membrane has several different kind of models, as most common, widely accepted as well this way, is the fluid model, as you're going to see it here, guys. The mosaic model is built of lipids, also proteins. The fluid part of the lipids. The lipids are constantly moving on the surface of the membrane, as you're going to imagine them like the water lakes, always moving. The most common population of lipids, guys, or actually which are present today, are phospholipids. Phospholipids, as we're going to see here, are structured as follows. Two parts, a tail and the head. So the head is hydrophilic and usually orientated toward water. The tails are hydrophobic. They're usually away from the water. So the structure of the membrane, guys, since we have this component of the phospholipids, is big phospholipid layer. This means that the heads are actually orientated to the surfaces, outside or inside of the cells, and then the tail are between the heads, so they're located in the center. The other molecules, lipid molecules we can observe in the cell membrane, are cholesterol. Cholesterol is the plasticity of the cells, guys. And this means the plasticity of the membrane, sorry. This means that the membrane can freeze in lower temperatures or increase the temperature of degradation or destruction of the membrane. Let's say some functions of the membrane. First thing what we need to know is that the membrane defines the shape of the cell. Also prevent a environment within the cell. Okay. So for example, there was not such ability of the cells. When you go to the guys, since there is no water, the cells will evaporate almost all of the water in order to balance the system. So when you go to swim in the river or lake, since there is a lot of, the cells will burst because there's going to be a lot of water going inside. Therefore, the cells have a special ability, you're going to understand, to allow specific things to go in and to go out. So this means that it's a unique environment which can highly differ from the outside environment. This is due to the fact that cells regulate what goes in and what goes out of the cells. This is one of the features. The only thing actually, guys, can pass freely through the membrane, those are the gases, the small non-polar molecules which can easily squeeze in between the phospholipids. All the rest they usually pass through proteins. We're going to talk about some protein channels. The cell membrane is used by the other cells to communicate to each other. They can exchange signals, also they can important events within the cells or neighboring cells, guys. Also program cell that is triggered that way too. Also the cell membrane is used to identify a is part of the body. Since it expresses several different receptors here, as you're going to see, and the combination of each of these receptors with the other one gives you the kind of immunogenic profile which is recognized by the cell, the immune system cell, and not attacked the cells of the, otherwise they get attacked quite a lot. Good. Continue on. The second component of the cell membrane are the proteins. As we said here, this is the mosaic part of the cell. So what is typical for the proteins? We're going to see the which can be kind of roughly be classified as two groups. We're going to see transmembrane protein, which also include the integral membrane proteins, and also we're going to see peripheral proteins. Transmembrane proteins, guys, are the one which is located on the surface of cells. They start from one part of the surface of the the membrane, and they pass through the whole membrane and on the other side. So most commonly, those are going to be channels, but they're not limited only to channels. Also here, some receptors can be transmembrane, guys. Also, some anchoring proteins can be and stuff like that. Go back to the protein channels. Protein channels are very important, guys, because everything else, part of gases, can pass through this one. So water usually pass through such structures. Ions, cations, anions pass through this surface. Big molecules like glucose, fructose also pass through such kind of surfaces, guys. So it's important to know that this one usually it's linked to an active, which means we energy in order to transport this kind of molecules, guys. That's why this transportation is very limited due to the use of energy. So the other proteins we're going to see are peripheral proteins. As you're going to imagine, they're usually located on the periphery of something. Either it's going to be inner membrane, inner part of the membrane, or outer part of the membrane. Those proteins are located only on the periphery. They don't pass through the whole membrane, guys. Typically for them, the one which are actually inner part of the membrane, inner periphery, this way, this can be several different types. For example, proteins located there can be enzymes, proteins, can be also some receptors, internal receptors, guys. The one guys normally are also receptors. The rare cases can be enzymes, but most of the time, in order the other molecules to bond to it, which are kind of glycoproteins, lipoproteins, and stuff like that, the stuff which used to form the glycocalyx. So the glycocalyx, guys, is very important outside of the cell membrane, which is a lot of, as we said before, glycoproteins, lipoproteins, also glycan chains we can find there, and plasma molecules. Imagine like surrounding the whole cell, guys. This glucose has several important functions. Most important for sure is the presence of glycans, which actually have ability to bind water, so the cells in the vicinity become water, which can use when it needs. Another example is that the glycoproteins and also proteins, which you're going to see on the top, something like a cloud, as I said before, which actually doesn't give an easy access to bacteria or fungi to the cells. So they need to pass through this kind of to access the cell, guys. And also, we're going to see some receptors surface which are getting recognized by the immune competent cells, so they can attack the cells if it's not presented in the body, or they will kind of pass by. Okay, this is about the cell membrane, guys. Now let's talk a little bit about the structure we're going to see on the cell membrane. In general, we separate them in two different types. We're going to see kind of flagellums, which are cilia, kinocilia, and we're going to see structures which we call microvilli. So the cilia, let's start first with one. The cilia, guys, is usually bigger than the microvilli, as you're going to see it here on this picture. Here is the microvilli, this is the cilia. It's bigger and thicker. So the cilia in general, it's a structure which can actively move the substrates, substance or substrates on the surface of the cells. So what is the structure in general? We're going to look from the cilia from the perspective of outside and inside of the cells. There are two distinct parts, guys. The inside of the cells is formation of the basal body. The basal body resembles the structure of this centriola, as it's formed of 9 triplicates of proteins, as you're going to see it here. Outside the cell, the structure changes significantly, guys, and it's 9 triplicates turns to 9 duplicates of proteins. But here, more importantly, we're going to see the microtubules in the center, two microtubules in the center, which actually are very important for the active movements of the structure. So thanks to this microtubule in the center, it's actively moving stuff, of course, using energy, as you're going to imagine. Sub-variant of this one is called stereocilia, which is way longer, as you're going to see it on this picture, compared to cilia. Stereocilia is typically found into the inner ear, guys, when the stereocilia into the neuronal impulses, as you're going to hear this way. The other variant of the membrane structures is called microvilli. Compared to the cilia, guys, microvilli not on the surface of the cells. They can still move, but this is going to be a passive movement. What is their function? Is the membrane surface in order to have a better absorption in the cells. So we're going to see here that the structure of the microvilli is completely different than the one of the cilia. This one doesn't have any kind of triplicates and duplicates. Here this is like normal micro. So the villi are built commonly from the microfilaments, which are extension of the cytoskeleton of the cells, guys. As you're going to see it here, we have actin and myosin filaments. Okay, we're going to look now for the microvilli, and we're going to look for the microvilli into the small intestine and most likely into the duodenum, guys. From the last time, you remember that the small intestine has very interesting structures which we call the villi, and those villi, guys, if you remember, have several different structures. On the top of it, we have actually 4 different layers. We have tunica mucosa, tunica varis, and tunica serosa, usually. So tunica mucosa, or the epithelium, guys, it has 3 more layers. As on the top one is located the tunica lamina epitheliaris, the epithelium. Underneath is the basal membrane. We have also that afterwards we're going to look with you for the muscularis mucosa. So with you again, we were going to focus on this tunica mucosa, and for the this is your the one for the cortex, the one epithelial cells, guys. We have a line, very pronounced line effect here. The surface brush the microvilli, which increase the absorption of the water can be stuff like that. Continue. Cell and stomach. Going to talk about matrix structures. We're going to see adhesions, or like focal adhesions, and of course hemidesmosomes, guys. Let's start first with the tight junctions. What are the tight junctions? First of all, guys, good to know is that the tight junctions are usually located in the, on the top of the cells, or we call it apical of the cells, guys. The importance of the tight junctions is that they have a close connections of the 2 membranes, guys, as you're going to see it here, which prevents going in between the cells. This is very important because this stuff which can go in between the cells can start to rot, guys, and this way can kill. Here, how the one tight junctions looks like. You're going to see that the 2 cells looks like needed to each other, guys. Very important to remember is that the tight junctions are also called zonal occludence. This is due to that the most important protein which is used for formation of the junctions is called occludin, guys. Occludin. Of other types of proteins, serines, catenins also can be actin included here. This doesn't matter. Now, tight junctions are the first one you can observe. Then somewhere around the middle of the cells, guys, we're going to see the other one, which we call anchoring junctions in general. Anchoring junctions can be separated into different subcategories like desmosomes and adhesion junctions. What's the difference? Well, both of the protein forms them. It's called catenin. What's the difference then? So since it has the same protein, there should be some difference how we can recognize them. Well, there is a difference comes from the stuff to which these catenin proteins are bind to the interfilaments, like for example, then we talk about desmosomes. Those are very robust structures, very thick one, because intermediate filaments are actually pretty thick. We're going to talk about these structures next week with you when we talk about cytoskeleton. The other thing we're going to do is the adhesion junctions are apparently bind to the actin, and actin is also part of the cytoskeleton, but it's called filaments there. Right. That's the difference to what they bound. When they bound to intermediate filaments, then we're going to call them desmosomes. When they bound to the actin filaments, then we're going to call them adherent junctions. Now, there is variations of binding of the extracellular matrix, and here we're going to see the hemidesmosomes. Usually, adhesion to the extracellular matrix is mediated by special protein receptors, which are called integrins. Integrins are heterodimers from one alpha and one beta subunit, which has the affinity to specific extracellular matrix, collagen, intronectin, fibronectin, and so on, guys. You're going to be surprised to learn they can also assess another cell. So you have cell-to-cell contact also mediated by integrins. But this is not important. So in this case, guys, you see that integrins mediates the link of the cells to the basal membrane. So when link to the keratins, as you're going to see here, or to the intermediates, then we talk about hemidesmosomes. But of course, integrins, as you're going to see here, also can be bind to the filaments, as you're going to imagine. And then when they're actin filaments, then we call them focal adhesions. Focal adhesions usually form very strong connections, guys, as they actually tend to form a very thick actin stress fibers, which are very difficult to destroy. The final cell-to-cell interactions we're going to talk with you is about gap junctions. Gap junctions are formed with unique, which we call connexins. Connexin monomers, guys, are usually and form this structure which we call connexon, as you're going to see it here. This one is a connexon. The connexon here forms one is closed one, as you're going to see here, but the other one is open one. The opening of the connexon happens due to the fact that we have presence of energy. Then the 6 connexins kind of shift a little bit, and then they open a hole, which is common, which kind of we have direct interactions between the both cytoplasms of the cells, guys. So through this kind of holes can pass only several differences. So of course, DNA, RNA cannot pass. Organelles also cannot pass. Ribosomes cannot pass through it, guys. But what can pass is like air, water, and of course, signaling molecules. This is very important because signaling molecules we're going to study with you are necessary to synchronize the muscle contraction of tissues like or internal, like smooth muscle cells, guys. Those cells have such connections with the gaps. Okay. Unfortunately, we cannot see these cell connections with the naked eye, with our microscope. Therefore, we need to limit ourselves with another type of connections we're going to talk about. Those are called interdigitational junctions or interdigitations, we call it also. How it's recognized, how they look So take one of the hand and the second one, then put the thing between each other, guys. You're going to see how the interdigitation looks like. So interdigitations, we are going to look with you in a special tissue called omentum ions, or the greater omentum, guys. Greater omentum is a special tissue, very thin one, which starts from the curvatures of the stomach, guys, and covers the internal structure of the belly. So it covers the belly on the top. So the guts and all the other stuff which are located into the abdominal part of the humans. Forms this structure are simple squamous epithelial cells. They're very thin ones, guys, but they have a very strong connection thanks to this interdigitation. This you can see here that the interdigitations looks like suturing two cells together. So those cells we're going to observe with you guys, those are simple squamous cells. But when we look from the top, guys, they look like this, very random shapes, guys. Their cells, if they don't have this connection, guys, will be easily overcoming the tissue, so it's going to be rupturing all the time, so our guts will spill. This doesn't happen thanks to strong interdigitations. Try to zoom in a little bit and try to find you some interdigitations here in this picture. For example, this region here, one you actually mark here. This one, also the thickening of this here, this is indicating the presence of interdigitations, guys. As I said before, this is very important one because it gives you the tissues the resistance towards the pushing, so they can resist a lot of pressure, guys. So with this one, we finished talking about the membranes. Now we'll continue, guys, talking a little bit about the ribosomes. So first of all, ribosomes, guys, as you know, they're important protein synthesis, guys, the process which we call translation. Also, ribosomes, guys, are usually built of units, as you're going to see them here, small and large subunit. In order to form the ribosomes, though, the two subunits need to bind together. It's a magnesium ion. This is one which has ribosomes. The mRNA can go inside and start the production of the proteins. Initially, ribosomes are assembled nucleus, guys. Surprisingly, they are going to be in the nucleus, and everywhere in the nucleus in a specific part which we call nucleolus. After the initial ribosomes, guys, they actually pass through this nuclear process, the one we're going to talk about with you. They pass through it, goes into the cytoplasm. When they enter the cytoplasm, the subunits, guys, but they can also be shipped to another part of the cells. Normally, we recognize two different types of ribosomes, guys. One ribosome which is in the cytosol, and then the other one which are of the endoplasmatic reticulum, or we also call the membrane bound. The free ribosomes, guys, are very interesting because a lot of proteins which one needs. So apparently these free ribosomes produce the proteins of the cells. Those can be structural proteins, they can be some enzymes and stuff like that, guys. Those are produced all in the free ribosomes. When the demand of some proteins is high, this means that several ribosomes can bind to the same mRNA, guys, and starting to produce the protein in a big amount. Such structure is called polyribosomes or polysomes, guys, as you're going to see them here, like this. This allows you that the protein which is produced is in a high amount for a very short time period. The second type of ribosomes, as I said before, are the membrane-bound or the ER-bound, the endoplasmatic reticulum-bound. As you're going to imagine by the name, they are located on the surface of the endoplasmatic reticulum. As when they bind to this surface, they look like a granules on the top of it. That's why this endoplasmatic reticulum is called granular plasmatic reticulum. We also call it rough endoplasmatic reticulum, guys. So the rough endoplasmatic reticulum produce also proteins, but these proteins which are produced, guys, they go inside the endoplasmatic reticulum. They don't stay in the cells, but they are rather used for the export. Such proteins can be collagens, can be structural proteins of the extracellular matrix, collagen, fibronectin, also glucosaminoglycans and stuff like that. We're going to show you how this process actually happens in just in a few minutes. But are you going to recognize that we don't have only the rough endoplasmatic reticulum, but we also have smooth endoplasmatic reticulum here. So the function of the smooth endoplasmatic reticulum obviously is not producing proteins, right? Otherwise, the ribosomes also on the smooth endoplasmatic reticulum. So the smooth endoplasmatic reticulum guys lipids. This is the place where the lipids integrate, guys. Also can be limited amount of carbohydrate. But this is not everything what the smooth endoplasmatic reticulum doing, guys. Smooth endoplasmatic reticulum can be storage. It actually stores the calcium ions into the muscle cells. Place when we get to the muscles to talk about this smooth endoplasmatic reticulum, and then we're going to call them there sarcoplasmic reticulum, guys. Sarcoplasmic because going to see typical muscle structure. Okay, then what else the smooth endoplasmatic reticulum can do? Well, it can locate the toxic component. For example, it's very important of the cell stuff. So this in general is what we need to talk a little bit about the endoplasmatic reticulum. Now we're going to see the slide we were supposed to see here. So the slide is medulla spinalis. Again, medulla spinalis we saw also last time. Though we're going to get with crysouvulate, or we're also going to call it anisostem. So I'm going to concentrate again onto the anterior part or anterior horn of the medulla spinalis, guys, as we're going to look into these cells which we called multoneurons. You remember for the last time that those cells, we said they are multipolar or starlight cells. Now we're going to dig into starlight cells and we're going to see that with the crysouvulates, we're going to observe staining of the nucleus, but not the whole nucleus as you're going to see it here, just a specific part of the nucleus which gets tied. This is the nucleus. Also, we're going to a part outside of the nucleus staying kind of blue ball actually of this rough endoplasmatic reticulum. So, beginning with the crysouvulate, to observe production of the nucleus, guys, and we're going to also the location of the membrane-bound nucleus and the top of the rough endoplasmatic reticulum. These structures we call missile bodies, guys. That's why I write that missile bodies equals the granular endoplasmatic reticulum. Okay, with this one we finish the first patient, and then we move for the first part of the second presentation, which we're going to talk about mitochondria, nucleus. As I said before, we will leave for the next first the structure of the cell. This is the animal cell, guys. So far we were talking about cell membrane, as you're going to see. We talk about reticulum and ribosomes. We left talk about mitochondria, nucleus, Golgi apparatus, lysosomes, centrosomes. We're going to talk about microtubules. This is skeleton. We're going to talk about next week. Now we're going to be focused on mitochondria, lysosomes, and Golgi apparatus. Of course, starting with mitochondria, guys, is a very interesting area. This is the place where the energy is used in the cells. As you're going to know, this is very important. The definition is that the cell can produce its own energy. It happens here. We're going to see that mitochondria two-membrane organelle. This means it has external and internal membrane, guys, or outer inner membrane also, if you want to call it. In between the two membranes, we're going to see the membrane space. It's a very important one. We're going to see why in a bit. Now, the mitochondria has a lot of pores, which we call purines. Two kind of pores can pass several different things. The most important thing which is passing, guys, is called pyruvate. So the internal membrane, guys, it's way more bigger than the internal one you're going to see here. That's why this one is kind of have some curves or imaginations of the membrane, which we call cristis. On the cristis, we're going to notice a special enzyme called ATPase synthesis, guys. The matrix of the membrane is located in the inner membrane, guys. It's a liquid which is in which we see the presence of DNA. This is the only other place of the cells we can see the own DNA. We're going to see RNA and we're going to see ribosomes. This means that this can self-replicate, guys, without needing of the cells to divide. Very importantly one. Now, within the matrix, we can also find a special cycle, which we call the Krebs cycle. Here is how the Krebs cycle is formed. So the pyruvate, which comes out from the outside, from the cytoplasm, guys, when it enters inside the mitochondria, start to be destroyed. This is a process, you know, typical of the organisms, which is linked to the production of energy. So the pyruvate is normally destroyed to 3 molecules of carbon dioxide, and then we have, I think, 2 molecules of water, if I maybe iron. But usually what's happened, leftovers of the carbon dioxide and water. During the process, though, on several occasions, you have the protons. These protons are usually catch up by special enzymes like NAT and FAD, and they are transported into intermembrane space. Then the protons which are located in the intermembrane space, because their gradient is higher than inside the matrix, guys, try to go into and balance the equation and then actually go in a species which we call ATPase synthesis, which moderates the transition of the 2 molecules of protons to actually link them to the oxygen, and then formation of the water can occur. But during this, if this process is not controlled one, usually it's linked to release of huge amount of energy to kind of bomb the cell, guys. Therefore, during this protons are binding to the oxygen, guys, and the formation of water occurs. The released energy is used by the ATPase synthesis to phosphorylate ADP to ATP, guys, as the ATP is the source of the energy within the cells. Where we are going to see with you mitochondria, we are going to put you in the or the same is unique one we're going to see with you guys in the end. It's iron hematoxylin. Iron hematoxylin, guys, thanks to the hematoxylin in the kind of black cover, and then takes the tissue in the black. Look for the nephrons, structural units, the major structural units of the kidneys, guys. Nephrons has the following structure. We have a Bowman capsule, which engulfs completely the device. And the core, you know, is kind of bow of capillary, which functions to increase the surface to which the blood can exchange stuff glomerule. It can go through from the blood to the glomerule, guys. Can toxic materials like picobilin, stuff like that, can also excrete things, and water is the very important thing which actually deliver to these kind of structures. What's happened with products which are released first, they pass the distal here, proximal tube, sorry, then pass through the loop of Henle, goes to the distal points, they're collecting, which release them in the brain or pelvis, and from there on, they can be fixed from the body. Important things here to remember is there is a reason why these nephrons are so long. They are so long because they usually storage for the water in the bodies, guys. The excessive amount of water is usually deposited into these kind of structures, but it's not discarded right. It stays there. That's why we have these big structures. Why? Because if the body needs water, first thing is kind of resolve back the water from the kind of structures of the neck. This happens due to the fact that a lot of blood kind of engulfing from guys yet also here. So what you're going to observe with you guys? We're going to observe here the tubes. This is a proximal tube. What you're going to see is that the tube, guys, has a lumen. This is the lumen of the tube. At the moment, the lumen is open, so it means that some kind of primary urine goes through. So those are the cells. I'm going to show that these cells has a circular nucleus. Do you see them? So when the circular nucleus, I'm going to draw them. Those cells have, we know that they have a cuboidal shape. I'm going to put them a little bit like this, you know, understand what we're looking for. Okay, those cells are cuboidal cells. Guys, this is the lumen. Usually the lumen is closed. Here is the circular shape of the nucleus. You see it? There, circular nucleus here. Also here on the other side. So, where we're going to look for you for the mitochondria. Look at this one. This cuboidal, guys, on the bottom has something like a stripes, black stripes. These structures here. The black stripes, which will be very close to the basal membrane, guys, are supposed to be the mitochondria. And here is your mitochondria. I'm not very good in drawings. Anyway, so this is the mitochondria. This is what I also see with you guys today. The presence of the mitochondria into the distal. Why do we see their mitochondria? Well, because, as I said before, when the water passes through the membrane, it's an active process. So it's energy. And here the water goes inside the cells and outside of the cells, guys. So it will require double the amount of energy. So therefore it requires a lot of energy to function properly, guys. Okay, this is about the mitochondria. Let's talk a little bit about the Golgi now. We are getting to the part, and first we will start with a special which we call exocytosis. What is exocytosis, guys? Exocytosis is a process of which the cells export stuff outside. This happens, that usually starts from the endoplasmic reticulum. Doesn't matter whether it's rough or it's a smooth endoplasmic reticulum, guys. It starts from there, then delivered to the Golgi apparatus, thanks to the transport vesicles transport stuff to the Golgi apparatus. After that, things get sorted, get out, and then can be stored in the membrane in order to exit this pathway. So when they send to the membrane, guys, usually the proteins which are released, you're going to see they are kind of smaller, then get assembled afterward. This one will teach in extensive amount when you get with you to the connective tissue. Then we're going to talk a little bit about the reason of exocytosis and what actually came to exocytosis. Right, as you see here, guys, Golgi apparatus has a key function. It's actually a key element of the exocytosis. Golgi apparatus, guys. Golgi apparatus, as you're going to see here, it's a membrane organ, guys. We call them also dictyosomes. So these cisternae, they can vary, but usually are between 10 to 12. They have clear differences, guys. We have part which comes to the transport, which is called cis part, and we have parts from which are released the secretory vesicles, which are called trans part, guys. So the cis part usually is orientated towards the nucleus or endoplasmatic reticulum. The trans part is usually oriented towards the cell membrane, guys. As I said before, in the cis part comes the transport vesicles. From the trans part get out secondary vesicles, that cells which are also keep is the function to and sort needs to sort them out. Them guys. You can observe the shape, it can become a global structures, or also you have also the third type structures. They can attach ions to them, or they can be selected due to the special tag molecules in the end. You can actually save the kind of ship them in a specific locations afterwards. So you see a lot of things can happen in the Golgi apparatus. What else I want to tell you guys is that actually the apparatus is very important in a special process which is actually called membrane recycling. So membrane recycling, I don't know whether you're going to touch this subject on the lectures, guys, but it's essential for, because this helps that the cell is changed and renewed, therefore the cell experience any kind of features. So what you're going to see here is that the importance of the Golgi apparatus not only to deliver stuff outside of the cells thanks to these secretory vesicles, but also the secretory vesicles are used to kind of deliver the new membrane to the surface of the cell. Exocytosis is the opposite of endocytosis process, guys. In the process of endocytosis, we actually, the cells get stuff from out and put it inside, guys, as this way kind of lowering down the amount of membrane on the surface. So imagine if there's only an endocytosis process, you're going to see that the cells will eventually shrinks a lot, and then all the organelles are kind of going to be pushed in the center and then squashed there. And therefore, for sure not going to be a space for functions of the bodies, of the organelles. So therefore, we need to find the balance between endo and exocytosis, guys, as the exocytosis is the opposite of the endocytosis. Here, you're going to see how the parts of the membrane can be replaced. Keep in mind that sometimes the membrane can be damaged itself. So, for example, some of the phospholipid layer can be destroyed. So, it's going to be only one layer of phospholipids. Then this structure usually is kind of engulfed and then put inside, and then the Golgi apparatus releases a new molecule, like a secretory vesicle will go and fuse with the membrane. So therefore, we have replenished the damaged membrane. Now, let's talk a little bit about the other structures which can be released from the apparatus. As I said before, the lysosomes. Lysosomes, also peroxisomes, guys, are very important also. Compared to the peroxisomes are having hydrolytic and peroxisomes, as you're going to imagine by the name, has only hydrogen peroxidases. Those are enzymes which can destroy the hydrogen peroxide, guys. Now, let's concentrate back. Lysosomes, guys, are simple membrane or single membrane organelles. One membrane, you're going to see, they're not so big one. What is significant that the membrane is a special one. First of all, directly underneath the membrane, we're going to see this glycoprotein called that separates the membrane of the lysosomes. This glycoprotein, guys, usually is the presence of inhibitors of the enzymes, the ones which are located in the lysosomal matrix. Furthermore, we go that on the membrane we have a lot of proton pumps. Proton pumps guys put these protons which lowers down the pH. So the pH of this lysosomal matrix goes something between 2 and 5 pH. The cytoplasm is between 7.4. This is very important because there is a leakage in the lysosome. The hydrolytic enzymes which are inside will not completely destroy the cell. Because in the different pH, go out in the neutral pH, they will get inhibited. Now, tell you which kind of hydrolytic enzymes here you're going to see them. Several different classes. Proteases, lipases, amylases, nucleases, phosphorescent monoesterase. The guys usually cut the phosphatic groups, the phosphate groups. Nucleases, imagine, destroys the nucleic acid to monomers like cytosine, the amylase to destroy the carbohydrates, guys. Those normally, but they destroy them to the monomers like glucose and stuff like this. They destroy the lipids, but not completely, but destroy them to phospholipids, cholesterol, for example, stuff like that. Proteases destroys the protoamino acids. So what you're going to see here is that the whole proteins here, the one which actually located this kind of hydrolytic enzymes, they don't completely disseminate the structures to carbon atoms, for example, they cut them to monomers. Therefore, you're going to imagine that lysosomes, guys, are the closest to cell-feeding you can imagine. Is usually cells get stuff from outside. I'm going to talk a little bit about afterwards, and destroys it to monomers, which can be used in kind of building processes, in the anabolytic processes in the cells, guys, right away, can include them right away. So lysosomes can be two different types, guys. Lysosomes, the one which are just expressed by are called primary lysosomes, as you're going to, sorry, not nucleus, but the Golgi apparatus. They're called primary lysosomes. And of course, we have a secondary lysosomes. Secondary lysosomes, guys, can be formed by a different way. The most common way you can find them are the one which are fusing with the endosomes. And here is the endocytosis, guys. Process. Endosomes are usually formed when the cells find some parts outside which likes to include. So we can have some parts of the membrane of the cells, receptors, and all. When the cells, sometimes they leave some traces behind them, which can be the cell, the other cell used, for example, the receptors on the surface. So it will move and get all structure by getting put inside the cytoplasm, usually inside the brain, and then transport. This is called endosomes normally. The primary lysosomes the cells recognizing some bacteria or virus on the surface, then include protein, also wrapped in the membrane, guys, fuse it with that. The most severe case of cells, secondary lysosomes are due to cell starvation. This process occurs after huge starvation. The cells starting to destroy organisms by them in this kind of autophagosomes, as you're going to see here. Normally, here the first goes the mitochondria because they have no glucose. This means that they. So after secondary lysosomes, guys, we said here that the stuff are completely destroyed to the monomers, and then the cells get out of the monomers. What's left over, guys, it is some residual bodies. We're going to talk about this one next week with you. The residual bodies are normally kind of structures, pigments or crystal structures which cannot be destroyed by the enzymes here. Right. This is about mesozoos, guys. Now let's talk a little bit about the Golgi. Where we're going to see the Golgi with you? Golgi, we're going to put you in a special structure which is called ganglion spinale. Here is the ganglion spinale, as you're going to see. What's typical for it, guys, is very close to the spinale. It's the one we already observed today. But in this ganglion spinale are located the bodies of the sensory neurons. How this looks like when it's cut? This is the ganglion spinale stained with the silver impregnation, guys. What we're going to see here is that in some special areas we're going to see concentration of the staining within these neurons, which we call neurons. Usually the place we're going to go now. Let's talk a little bit about the pseudoneuron neurons. You're going to see that neurons, guys, can be several different types. I'm going to draw you some of this one. Okay, so unipolar neurons are this one, the one which has only one protrusion. Bipolar, which has two protrusions, usually they are on the opposite side of the cells, and multipolar neurons are the one which has several different protrusions. It's already said on last week, which we remember when we talk about medulla spinalis. Okay, so where is the pseudoneuron it's in, guys? This is your pseudoneuron. So it starts usually with one protrusion, that's why it's unipolar, but then it splits into, therefore forming like something like structure like a T-shape, as you're going to see. That's why these neurons are also called T-shaped neurons here. So the pseudoneuron neurons are pretty big one, guys. You're going to see it around them, you're going to see a bunch of nucleus, which are actually of these glial cells. We're going to talk with you about those are called satellite cells. But at the moment you think in that one, we're more interested in the structure of these pseudoneural neurons. What you're going to see on the first glance, guys, is that this one is the cell. See, this one is the cell. The cell nucleus is not stained at all. This is typical for this staining. What is stained is just part of the underneath the nose, which is for the membrane. In this dark structure here, also here, and this one here, we're going to see the Golgi apparatus. So by staining the cellular neurons with the silver impregnation, we will be able to see the location of the Golgi apparatus within the cell. Okay, with this one, I will finish the part for today. Just let me see here to write down the names, not that I missed someone. Rifa. So Rifa is not here, I think. Okay, so if you have questions, put them in the chat, guys. If not, I wish a very nice week. Hopefully see you soon here. I don't know, is there someone in Bulgaria from you yet or is here yet?